Hello class and welcome to week 6, part 2. Thought I'd review what we covered in part 1, where we're talking about the laws of thermodynamics. We can picture this stack of cups where this stack is going to have more order, it's more complex certainly, there's a lot more organization, and there's actually more energy in it, but it's less stable. All those cups on the floor are certainly more stable, but now they have less energy, they're less organized, there's less order, and they're certainly less complicated. And we went through a lot of examples of thinking about how order and energy and stability are all related. So thinking about that and thinking about two rocks that you saw, what happened with that energy? You had the rock that had a lot of energy up here, was released and fell down and hit this rock. Then this rock moved up. We had that exergonic reaction where the energy was released, and we had an endergonic reaction, that ball moving up that hill. So what happened was that these reactions were coupled. So that exergonic reaction used energy to drive that endergonic, pushing that rock up the hill reaction. And this is exactly what we're doing all the time in our bodies. We're coupling reactions that take energy with reactions that release energy. And how we do this is through ATP, through adenosine triphosphate, which you recognize from thinking about that nucleotide. So we saw, and we already talked about this a little bit, that what our body does is it makes and breaks this bond over and over again, adding a third phosphate group to those two phosphate groups that are already present, along with, of course, that nitrogenous base and that ribose sugar. When you have those three ATP and you release one of the phosphate, that release is energy. That's an exergonic reaction. When we build that third ATP, that's going to take energy. Again, I am expecting that you've already looked through these PowerPoints, and I'm not going to go over every single slide. But what we're talking about are these coupled reactions of taking an exergonic reaction, releasing energy, and releasing energy, coupling it to do the work that's needed for that endergonic reaction, so that the exergonic reaction provides the energy for that endergonic reaction. You can kind of think about it like a gear moving, where you're getting one to move up while the other is already moving down. So going back to that idea of gasoline, and we can think about how we can couple a reaction. If you have 100 units of energy stored and potential energy in that gasoline, and you light it on fire and produce carbon dioxide and water, which are far more stable but have less potential energy because there's less bonds, there's less energy in those bonds, that's going to release 100 units of energy. If we think about moving a car that's stationary, that's not moving, up a hill, that's going to require 25 units of energy. So we can combine these, we can couple these in the burning of the gas, providing that energy to move the car. The first law of thermodynamics, energy isn't created, destroyed. The second law of thermodynamics is whenever we're doing these reactions, we're going to increase the enthalpy, increase the disorder, increase the heat, so to speak. In this case, most of that energy is going to heat. Coupling the reactions in our body, we can think about how do we get our muscles to contract? How can we lift up a heavy weight? We are using the energy stored in ATP, and we are coupling that with that muscle contraction. So the cells in our body are going to do chemical work, which we're going to talk about from moving chemical bonds. There's also transport work and mechanical work that is also being led by the hydrolysis of ATP. So here we can give yourself a little quiz. Label these reactions. Which one is exergonic? Which one is endergonic? I'll assume you can have paused this because I'm going to go right into saying it's endergonic to build up ATP. It is exergonic. You are releasing energy when you're breaking down glucose into that simpler, more stable carbon dioxide and water. So this is going to release energy that's providing the energy to make ATP. Equally, you'll need to pause here, label those reactions. You have that exergonic reaction of releasing that third phosphate to become ADP, releasing energy. And in this case, this C plus D equaling A to B would then inherently be the endargonic reaction. So this is what we mean by coupling. So again, more questions to check yourself with the answers. So another question we can ask ourselves is, looking at this reaction, which hopefully you're going to start to recognize as what we're doing in aerobic respiration, taking sugar and oxygen and producing carbon dioxide and water, we can ask ourselves, which is more stable? Which has less energy? Hopefully everyone thought carbon dioxide and water, less stable, less potential energy, but more enthalpy. So will this reaction occur spontaneously if we have higher free energy and lower free energy and we're releasing it? Or does sugar just spontaneously combust? No, of course not. And why is that? Because you first need to break the bonds that are holding glucose together. So we can think about those valence electrons, and they'd first have to be broken before they can get rearranged. And what we call that, the energy that's needed to have this reaction begin, is called the activation energy. So if we draw this, the real free energy diagrams, where we start with the energy of the reactant, so in this case ethanol, that was 10 kilocalories, we actually need to break down these bonds first. So there is going to be more energy that is going to be required to break those bonds, even if at the end you are going to have less energy in the ultimate products. There is this in-between state that is going to have a higher amount of energy required than even the energy in the reactants. It's this difference that's called the activation energy. So you ask yourself, where is there the greatest instability? Right there, where there is the highest amount of potential energy. You can ask yourself, which is more stable, the reactants or the products? It is the products that, of course, are more stable. They have less potential energy. They are the lower point on this graph. The highest point has the most energy. The lowest point has the lowest energy. So how can this reaction occur? What is going to be providing that activation energy? Well, one thing we know that can provide energy in a cell is heat. If these molecules are moving up around a lot more, eventually those bonds can get broken. So heat and increasing thermal reactions is possible. However, in our bodies, we don't need that because our proteins would fall apart if they get too hot, which we'll talk about again in a little bit. So what do our bodies use? Our bodies use ATP and enzymes to lower the activation energy needed for a reaction to occur. So we're going to learn about enzymes. Enzymes provide the physical place and the physical little room in the space to make reactions occur. And we're going to see an enzyme forms a complex with a substrate. That's what it's called, what binds an enzyme, and it produces a product. So enzymes lower the activation energy needed for a reaction to occur. So if we think about an endargonic reaction, you still can have an enzyme that is going to decrease the amount of activation energy needed for that reaction to occur. Either case, you still need an input of energy to make those products with higher free energy than the reactants, but an enzyme is going to lower the amount that's needed. You can watch that video. So again, some more terms. A catalyst is simply just a chemical reagent that speeds up a reaction. So an enzyme is a catalytic protein. We'll use this example that is going to narrow in on some of the key terms you need to know for enzyme reactions. So if you have sucrose right here, and you want to add water to bust it into its monomers, glucose and fructose, an enzyme called sucrase is going to do that. Quick little questions that you can ask yourself to make sure you're knowing the material and the level you need. What category of macromolecule is sucrose? Well, that's a carbohydrate. Sucrase is an enzyme. Words that end in ACE, A-C-A-S-E in biology, those are enzymes. So is this a catabolic or anabolic reaction? We are going to be breaking things down, and that's going to be catabolic. And this would be an example of a hydrolysis reaction using water. And we can label the substrate, we can label the enzyme, sucrase, and the product, glucose and fructose. So here we're going to go through in depth. So here we have an enzyme. There's a specific part of the enzyme that's called the active site. Now the active site is where the substrate binds. So here we have that sucrose substrate, and here you have it binding directly in that active site. Sucrase is going to use water, and it's going to break that bond between the glucose and the fructose, releasing the products. Now, when those products are released, the enzyme is exactly how it was when it started, which is the function of enzymes is not to be changed by reactions. In fact, enzymes do not change. They are just reused and reused and reused. In sum, enzymes lower the activation energy needed for a reaction to occur. Now here I wanted to draw everyone's attention to the fact that, as I said before, the active site is physically where that substrate bonds. For why we went on and on about proteins is that the structure of a protein is going to be directly linked to the substrate, to the reaction in which it's involved. Because there's that enzyme substrate complex that's formed, it's an exact fit. So each enzyme is going to be involved in a different reaction. And what those enzymes are doing is they're going to hold the substrates in a particular way, in a particular order, to make a reaction more favorable. They might bend things so that they're not quite fitting, or they might put things together, sort of acting like a matchmaker, bringing things together, or getting that wedge in there to break things apart. They create a particular microenvironment, which is going to facilitate that chemical reaction occurring. Other key terms to know is cofactors. Those are non-protein enzyme helpers. We're going to see in some enzymes later on that mostly it's metals. Coenzymes are organic cofactors, and what that means is that they help a reaction occur, but unlike enzymes, which do not change, which are not changed by a chemical reaction, coenzymes are. Coenzymes can be modified by the reaction. So now we need to remind ourselves of what are enzymes. Enzymes, of course, are proteins. And so proteins can denature or be made to be inactive when you're breaking down all the bonds in that tertiary structure. It doesn't even have to be all the bonds, just some of the bonds. When you break them down, that protein is going to fall apart and denature. It's not going to be active. So what are those things? That's temperature, pH, or salt can lead to denature. These are also the things that are going to affect how enzymes are functioning. So an enzyme is going to have an optimal temperature and an optimal pH for which it's going to have the right shape. So we can look at two different enzymes in this case, and we can see that there's one enzyme that is going to be most active at 37 degrees based on the fact that we have the rate of reaction is on our y-axis and temperature is on our x-axis. So as temperature increases up to about 37 degrees, that enzyme is becoming more active. After that point, there's a very sharp decrease. As that temperature increases, that protein is going to denature. Now, there's other enzymes that are going to function at a different optimal temperature. In this case, you have a bacteria that lives in very hot water. Its optimal temperature for reactions to occur in that enzyme are going to be much closer to 77 degrees Celsius. But of course, they have a limit too, and that protein can only get to some extreme temperature before it too will denature. So you can ask yourself, looking at this graph, thinking about the rate of reactions, where it's increasing as it gets higher and decreasing as it gets lower relative to pH, which would be found in a tissue of a human being or in your gastric juice in your stomach. Hopefully you answered pepsin with that extremely low pH that it's active at. That's going to be in your stomach, where your intestinal enzyme of trypsin, that's going to be in our normal body temperature, our normal body pH of around 8. So enzymes are regulated by temperature, by pH. They can also be regulated by inhibitors. So what is an inhibitor? It's exactly what the name suggests. It inhibits. So you have normal binding of that substrate binding into the active site of the enzyme in that induced fit, lock and key idea where it's fitting straight in there. But you can have a competitive inhibitor. A competitive inhibitor is, like in sports metaphors, it physically gets in the way. It is physically blocking that active site. So a competitive inhibitor blocks the active site. A non-competitive inhibitor is going to bind that enzyme and make that active site non-functional, but it's not going to physically be in the place blocking it. So a non-competitive inhibitor still is inhibiting this enzyme from cleaving this blue blob, but not because it's physically in the way. Another term for non-competitive inhibitor is an allosteric regulation. Allosteric regulation is when it's binding someplace other than the active site. So it's going to bind something other. So an allosteric. And this idea of allosteric regulation can actually come in the form of being an activator or in the form of an inhibitor. The inhibitor form, again, is an example of non-competitive binding, so it binds someplace else on the enzyme than the active site. In the active form, still binding someplace not the active site, but it binds in a way to make that reaction continue occur. So you can see in this case, that substrate is able to bind, and that activator is help keeping that shape. So all these ideas of how enzymes are controlled is really important if we go back to thinking about metabolism as all of the different pathways that are building up and breaking down our cells. So here we have that same picture that was in just block form earlier, but you can zoom in and so we can see that it's the regulation of enzymes that are actually going to be controlling all of those processes of metabolism. All those building up and breaking down are regulated based on enzymatic activity. And what we call this is feedback inhibition. This is a case where you have a metabolic pathway where you have reactant 1 with enzyme 1 producing an intermediate. That intermediate is get acted upon by a different enzyme because of course it's the shape of that enzyme, so a different substrate is going to have a different enzyme. And then this enzyme converts that intermediate to this orange intermediate. And then there's a third enzyme that's going to produce that final product. The way that all of our metabolic pathways can keep occurring and you don't have total craziness is the product at the end will often feed back and regulate the enzyme at the early part of that metabolic pathway, where the product will serve as an inhibitor for one of those first enzymes. So another same example, different idea. You have your first product A, you have enzyme 1, 2, 3, 4, 5 go through and produce a lot of that end product F. That end product F is going to physically bind enzyme 1 allosterically, so not in the active site, but it's going to prevent enzyme 1 from converting that first reactant A into B and therefore blocking that pathway because you have the end product that you want. So here's another good example where your end product isoleucine comes back and it's going to inhibit one of the first enzymes in a whole series of pathways. And again, as a reminder, our body is using all this energy for mechanical work to contract and move things, for chemical synthesis of building up all those macromolecules, and then of course to move things across the membrane and into various compartments. And it's enzymes that are needed to form or to break covalent bonds. So what we're doing in this chemical energy is we are making and breaking bonds. If you're making and breaking bonds, you're going to need energy to reduce that activation energy. So you can do a quick little quiz of yourself, and I've got all the answers, and then here you can see all the information that you are going to be expected to know.